It's the job of the urinary system to maintain the constancy and quality of our body fluids. The kidneys are the main organ that carries out this function. The urinary system functions with the skin and lungs in excretion. After the final urinary product is produced, it is collected and conveyed to the ureters, which conduct the urine to the bladder where it is temporarily stored, and then when convenient or socially acceptable it is released to the exterior via the urethra. The kidneys filter about 200 liters of fluid from the bloodstream daily. They guard and protect the quality of the blood and other body fluid compartments by removing toxins, metabolic wastes, excess ions and molecules which leave the body in the urine. In addition, they also return needed substances such as glucose and amino acids to the blood, regulate volume, blood pressure and maintain the proper acid-base balance pH. Other renal functions include gluconeogenesis during prolonged fasting. Gluconeogenesis is the formation of glucose from non-carbohydrate molecules. The kidneys also carry out several endocrine functions such as secreting erythropoietin, which stimulates red blood cells production, and metabolized vitamin D to its active form. In this unit, we will study renal physiology, how the renous kidneys maintain the constancy of the blood, regulate blood pressure, balance blood pH, and respond to changes in plasma osmolarity. Before beginning, review the anatomy of the urinary system. A few slides in this presentation review the anatomy of the kidney. Interactive Physiology has an anatomy review, as well as some great animations on different aspects of renal physiology. Each kidney is surrounded by a connective tissue capsule. Just inside the capsule is the renal cortex, the most superficial part of the kidney. The cortex contains numerous nephrons, which are the structural/function unit of the kidney, and renal columns, with our collections of ducts and tubes that extend between the renal pyramids of the medulla. The inner part of the kidney is the medulla. It contains 8 to 18 renal pyramids, so named because of their shape. The base of each renal pyramid faces the cortex, and the apex, known as the renal papilla, points toward the inner center of the kidney. The renal pelvis is part of the collecting system. It contains cup-shaped extensions called calyces. A minor calyx is found at the apex of each renal pyramid. It collects the drops of urine from the renal papilla. Several minor calyces fuse to form a major calyx, and the major calyces fuse, forming the renal pelvis. Between the calyces is a space called the renal sinus. Typically, this is filled with fat and is part of the protective and supportive coverings of the kidney. The medial aspect of the kidney is indented and called the hilus. The renal artery enters, renal vein and ureter leave the kidney at the hilus. Since the main job of the kidneys is to maintain the integrity of the blood, it's no surprise that the renal arteries deliver 1/4 of the total cardiac output. That equates to 1,200 milliliters of blood filtered each minute, or a total of 45 gallons/day. The renal artery enters the kidney at the hilus and then branches, forming 5 segmental arteries. Each of these divides, forming interlobar arteries which travel between the lobes of the kidney. A kidney lobe consists of a renal pyramid and its associated cortex. The interlobar artery arches over the bases of the medullary pyramids, where they are called arcuate arteries. Branching off of them are small arteries known as the cortical radiate or interlobular arteries. The cortical radiate arteries give rise to a number of short arterioles, each known as an afferent arteriole. The afferent arteriole forms a ball-shaped capillary bed called the glomerulus. What comes next is another exception to the usual circulation pattern. Instead of the glomerular capillaries being drained by a venule, the capillaries drain into another artery called the efferent arteriole. The efferent arteriole forms a second set of capillaries called the peritubular capillaries because they wrap around the renal tubule of the nephron. They lead into venules to the corticoradiate veins, arcuate vein, interlobar vein, segmental vein, and finally the renal vein, which returns the blood to the inferior vena cava. The structural functional unit of the kidney is a structure called a nephron. Each kidney contains over 1 million nephrons. The nephrons are microscopic, continuous tubes only 1 cell layer thick, but if stretched out and laid into the tubules would be 75 miles long. The sheer number of nephrons per kidney tells us that they play a crucial role in the body. As we will see, their design is what allows the kidney to pack enough nephrons in an organ the size of a fist to do the job. The kidneys are truly one of God's most creative pieces of work in the human body. Anyway, the nephron carries out 3 basic functions: filtration, reabsorption and secretion. The picture in the slide above shows a schematic diagram of where along the nephron each of these activities takes place. I want to define what is meant by a couple of the functional terms to avoid confusion later on. First, filtration is driven by hydrostatic pressure. In the body this is blood pressure. The pressure I is higher in the glomerular capillaries than any other capillary bed in the body. The pressure pushes fluid and certain materials in plasma through a filtration membrane. The fluid is collected and begins its journey down the tube, so to speak. Many of the filtered materials the body wants to keep, so they are reabsorbed from the filtrate. The process is called reabsorption because many of the materials in plasma were originally absorbed by capillaries in the intestinal tract. In reabsorption, materials are pulled out of the fluid and put back into the bloodstream. Secretion means that materials that need to leave the body that were not removed by initial filtration are taken out of the blood and are secreted into the fluid that will ultimately leave the body as urine. Each nephron consists of two main parts. The renal corpuscle is the site where blood plasma is filtered and the initial filtrate is formed. The filtrate will be modified and adjusted as it passes through the rest of the nephron. The renal tubule receives the filtrate and modifies it as it travels throughout its course. There are three anatomical and functional parts to the renal tubule. They are the proximal convoluted tubule, loop of Renal, and the distal convoluted tubule. Each nephron dumps into a collecting duct, shown in gray in the picture above. The collecting duct is not part of the nephron. Each collecting duct receives tubular fluid from multiple nephrons. The renal corpuscle is where filtration of blood takes place. It consists of two parts, the glomerulus, which is a tuft of capillaries derived from the afferent arteriole and hangs into a spherical structure made from epithelial cells called Bowman's capsule. Bowman's capsule is double-walled, it has an outer parietal layer which forms the globe into which the glomerular capillaries hang, and a visceral layer. The visceral layer is made up of specialized cells called podocytes, meaning literally, footed cells. The podocytes adhere closely to the endothelial cells of the glomerulus. The first part of the renal tubule is called the proximal convoluted tubule. Proximal because it is the part closest to the renal corpuscle where the filtrate is made. Convoluted because it wraps around and around, ensuring that there is enough surface area for reabsorption of materials. The microscopic anatomy of the PCT shows that the cells are specialized for absorption by the presence of microvilli, collectively forming a brush border. The PCT plunges into the medulla of the renal pyramid where it straightens out, forming the loop of Renal. The loop of Renal makes a sharp U-turn and heads back up into the renal cortex. The loop of Renal consists of a descending thick limb comprised of simple cuboidal epithelium, descending thin limb and descending thin limb, both made of simple squamous epithelium, then the ascending thick limb, which becomes the distal convoluted tubule. The DCT joins the collecting duct. There are two types of nephrons. About 85% of the nephrons are cortical nephrons because all of the renal tubule except for a small part of the loop of Renal resides in the cortex. The remaining 15% are specialized for conserving water. They are called juxtamedullary nephrons. Juxta is a prefix meaning next to, so the renal corpuscles and convoluted tubules are found close to the base of a renal pyramid. Juxtamedullary nephrons have extremely long loops of Renal that extend down the entire length of a renal pyramid. As alluded to previously, nephrons have two capillary beds. One, the glomerulus is derived from the afferent arteriole, the other capillary bed is derived from the efferent arteriole and forms the peritubular capillaries. One way you can remember what arteriole is going where is to refer to A for afferent and is for approaching. The afferent arteriole brings blood to the glomerulus. E is for efferent and is for exiting. The efferent arteriole takes blood from the glomerulus to the peritubular capillaries. The capillaries forming the glomerulus consist of loops that run in parallel. They are specialized for filtration because they are fenestrated. The peritubular capillaries cling to the renal tubules of cortical nephron. They are under extremely low blood pressure and are porous for reclaiming useful materials from the tubular fluid. Another specialization of the nephron capillary beds is that the peritubular capillaries of the juxtamedullary nephrons are connected to the vasa recta. The vasa recta, as the name indicates, are long straight vessels that parallel the long loops of Renal. The endothelial cells comprising the vasa recta have unique qualities not found anywhere else in the circulation. Each nephron is equipped with a structure called the juxtaglomerular apparatus or JGA. The JGA consists of two parts. The first is the macula densa, which simply means dense spot. The macula densa consists of a small area of tall, crowded cells in the distal convoluted tubule adjacent to the afferent arteriole. These cells are specialized as chemoreceptors, and they monitor and respond to changes in the concentration of sodium and chloride ions in the fluid reaching the DCT. The second component of the JGA are granular or JG cells. They are a small group of specialized smooth muscle cells found in a small part of the afferent arteriole just before it enters the renal corpuscle and forms the glomerulus. The JG cells are filled with granules of an enzyme called renin, which ultimately functions to maintain blood pressure and filtration rate of the kidneys. The JG or granular cells are able to sense the blood pressure in the afferent arteriole as it enters the glomerulus. Together, the components of the JGA play an important role in regulating blood pressure and maintaining a steady filtration rate. The gray cells between the glomerular capillaries are called mesangial cells. They are not part of the JGA but do play a role in regulating blood filtration rate. On the average, about 1,200 milliliters of blood per minute is filtered, and 125 milliliters of filtrate is formed per minute. This is equivalent to filtering your entire plasma volume over 60 times each day. Before we continue, we need to review the body fluid compartments and define the terms: filtrate, tubular fluid, urine. Filtrate contains everything found in blood plasma except proteins. By the time the filtrate enters the collecting ducts, most of its water, nutrients, and ions have been returned reabsorbed to the blood. Urine is what remains as the final output product. The kidneys process 180 liters 47 gallons of filtrate daily. Typically less than 1% 1.5 liters leaves the body as urine. Urine formation and adjustment of blood composition involves three processes: 1 glomerular filtration, 2 tubular reabsorption, 3 tubular secretion. The body fluid compartments include the interstitial fluid surrounding the nephron. This is frequently referred to as peritubular fluid, which means the fluid around the tubule. Blood plasma is the other fluid compartment that plays a role in renal physiology, since it is the quality of the blood that is the object of filtration, reabsorption and secretion processes. The first step in urine formation is filtration. Recall that filtration is a passive process driven by hydrostatic pressure, blood pressure, that forces fluids and small solutes through a filtration membrane. The filtration membrane, found in the renal corpuscles of nephrons, has a tremendously large surface area, and is thousands of times more permeable to water and solutes than membranes elsewhere in the body. The blood pressure entering the glomerular capillaries is much higher than the pressure of blood entering all other tissue capillary beds, about 55 mm Hg compared to 10 to 18 mm Hg. The renal corpuscle is the site of filtration, the first step in the production of urine. The renal corpuscle consists of two components: the glomerular capillaries, and Bowman's capsule. Bowman's capsule is double-walled, consisting of an outer part called the parietal layer, and the visceral layer comprised of specialized cells called podocytes, which adhere closely to the endothelial cells of Th. E. glomerulus. In the picture above, the podocytes are shown in tan, covering the left half of the glomerular capillaries. In life, the entire glomerulus is covered in podocytes. The parietal layer forms the capsule that collects the filtrate as it is produced. The podocytes and glomerular endothelial cells form the filtration membrane. The glomerular filtration membrane is comprised of three combined structures originating from the glomerular capillaries and podocytes. The endothelial cells comprising the glomerulus are fenestrated, meaning that they have pores. The visceral membrane of the glomerular Bowman's capsule is made up of podocytes. Both the glomerular capillaries and the visceral layer are epithelia, and so they are supported and attached to a basal lamina. However, the basal laminae of the two layers fuse, forming a thin membrane. In order to facilitate filtration, it is half as thick as having two separated basal laminae. Each of these three components is designed to permit certain size solutes to pass, and others to be retained. First, the fenestrations of the glomerular endothelial cells prevents the filtration of blood cells but allows all other components found in plasma to pass through. The fused basal laminae prevent filtration of the larger plasma proteins. Between the petzels of the podocytes are filtration slits that prevent filtration of medium-sized plasma proteins. Let's examine the filtration membrane more closely. The endothelium of the glomerular capillaries has pores fenestrated that are 50 to 100 nanometers in diameter 0.0000000005 m. The pores prevent blood cells from entering the filtrate. A basement membrane between the endothelium and visceral layer of Bowman's capsule prevents large proteins from entering filtrate. The visceral layer of Bowman's capsule podocytes have thousands of cytoplasmic extensions called pedicles which cover the basement membrane like interlaced fingers. They form filtration slits. The pedicles function to support the filtration membrane and to restrict passage of medium-sized proteins. Some materials can pass directly through the filtration membrane unimpeded. These include molecules smaller than 3 nanometers. 1 nanometer equals 0.0000000001 m and include glucose, water and amino acids. Clinical note: damage to the kidneys resulting from mechanical trauma, bacterial infection, circulating immune complexes, or exposure to metabolic poisons can result in a sudden increase in glomerular permeability. If severely damaged, plasma proteins proteinuria and blood cells hematuria can enter the capsular space and leave the body in the urine. This creates severe, wide-ranging physiological imbalances. As mentioned previously, the blood pressure entering the glomerular capillary bed 55 mm Hg is greater than the pressure in other capillary beds 18 mm Hg. Vascular resistance is what drives filtration of the blood. Recall the unique circulatory route of blood flow to nephrons. The afferent arteriole delivers blood to the glomerular capillaries which are drained, not by a venule, but by another arteriole called the efferent arteriole. The afferent arteriole is short but has a relatively large diameter. The efferent arteriole has a smaller diameter than the afferent arteriole. This difference in the luminal diameters of the inlet and outlet arterioles contributes to vascular resistance that produces the pressure for filtration. There are 3 different filtration pressures that determine the net filtration pressure. The net filtration pressure NFB is responsible for formation of the filtrate and involves forces acting at the glomerular capillary bed. Glomerular hydrostatic pressure GHB is the blood pressure in the glomerular capillaries. This pressure pushes water and solutes out of the plasma and through the filtration membrane. There are 2 other pressures that oppose GHB which serve to inhibit fluid loss from the glomerular capillaries. These filtration opposing forces are 1 colloid osmotic oncotic pressure of the glomerular blood, and 2 capsular hydrostatic pressure exerted by fluids in the glomerular capsule. The capsular hydrostatic pressure is the fluid pressure of the filtrate 15 mm Hg that is already in the capsular space. It opposes the GHB because it tends to push water and solutes back into the bloodstream. Colloid osmotic pressure also opposes GHB, but in this case it is the back pressure resulting from plasma proteins in the glomerular capillaries. Plasma becomes more concentrated as filtrate is formed. Fluid leaves plasma and enters Bowman's capsule, but proteins stay in the blood in the glomerular capillaries. Blood colloid. Idle osmotic pressure pulls water out of the filtrate in the capsular space and into the plasma, thereby opposing filtration. COP averages 25 mm Hg. The net filtration pressure NFB therefore equals the glomerular hydrostatic pressure, minus the sum of the 2 opposing pressures capsular hydrostatic and blood colloidal osmotic pressures. The average pressure entering the glomerular capillaries is 55 mm Hg minus the average capsular hydrostatic pressure 30 mm Hg plus the blood colloidal osmotic pressure 15 mm Hg. This equals a net filtration pressure of 10 mm Hg. This is the average pressure forcing water and solutes out of the glomerular capillaries and into the capsular space. Glomerular filtration rate GFR is a clinically important parameter and is measured routinely in patients. The GFR is the volume of filtrate formed each minute by the combined activity of all 2 million glomeruli of both kidneys. GFR in normal adults in both kidneys is 120 to 125 milliliters per minute. There are 3 main factors governing filtration rate at the capillary beds: 1 total surface area available for filtration, 2 filtration membrane permeability, and 3 the net filtration pressure. Because glomerular capillaries are permeable and have a huge combined surface area about that of the skin, 22 square meters, enormous amounts of filtrate can be produced even with the NFB being only 10 mm Hg. However, a drop in glomerular pressure of only 18% stops filtration altogether. Because the GFR is directly proportional to the NFB, Any change in any of the pressures acting at the filtration membrane GHB, sees HP, or POP changes both NFB and GFR. It is critical that the GFR be tightly regulated or kidney function would be severely impaired, which could lead to electrolyte and fluid imbalance, inadequate clearance of the blood of toxins or excess solutes, shift in blood pH and blood pressure. For example, by changing just one parameter like increasing systemic BP, if it weren't for tight regulatory mechanisms, then filtration would increase and the body could dehydrate, too much water would leave in the urine. Conversely, if systemic BP decreased, then filtration would slow or stop and the kidneys could not adjust the blood composition or volume. GFR is regulated by both intrinsic and extrinsic controls. These two types of controls serve two different and sometimes opposing needs. The kidneys require a relatively constant GFR in order to maintain extracellular homeostasis. The body as a whole needs to maintain blood pressure within specific limits, and therefore requires a consistent volume of blood. Intrinsic controls are also known as renal autoregulation because they act locally within the kidney to maintain GFR despite minor fluctuations in blood pressure that occur all the time. The extraneous controls act through organ systems outside the kidney, specifically the nervous and endocrine systems. Their goal is to regulate GFR to maintain systemic blood pressure should it get dangerously high or low. Note, in extreme changes of blood pressure, mean arterial pressure of less than 80 or greater than 180 mm Hg, extrinsic controls take precedence over intrinsic controls. FYI, normal, average BP for healthy 20-year-old adults is 127th of a millimeter Hg. Let's look at how the kidney self-regulates filtration pressures by adjusting its own resistance to blood flow in order to maintain a steady GFR. Renal autoregulation entails two types of controls, one a myogenic mechanism and two a tubuloglomerular feedback mechanism. For your information, Interactive Physiology has an excellent animation of these controls which will really help you understand how the mechanisms work. Let's begin with the myogenic mechanism. When GHB increases, the smooth muscle in the wall of the afferent arteriole is stretched. The smooth muscle cells respond to stretch by contracting, similar to stretching a rubber band. The more it is stretched, the more forcefully it snaps back. Therefore, increasing systemic blood pressure causes the afferent arterioles to constrict, which restricts blood flow into the glomerulus and prevents glomerular blood pressure from rising to damaging levels. Conversely, declining systemic blood pressure removes the stretch on the blood vessel wall, causing it to go slack. This causes the afferent arterioles to dilate, and more blood flows into the glomerular capillaries, thus raising glomerular hydrostatic pressure. In both situations, the myogenic response helps to maintain a normal and steady GFR despite fluctuations in systemic blood pressure. The second intrinsic control mechanism is the tubuloglomerular feedback mechanism, which involves the macula densa cells and the JG or granular cells of the juxtaglomerular apparatus. Recall that the macula densa consists of some closely packed, tall cells in a small area of the distal convoluted tubule where the tubule comes into close contact to the renal corpuscle. The cells of the macula densa are sensitive to changes in sodium chloride concentration of the tubular fluid in the distal convoluted tubule. In short, the sodium chloride concentration reaching the macula densa varies directly with glomerular filtration rate. In other words, when GFR increases, more filtrate is being produced faster, so there is insufficient time for the renal tubule to reabsorb NaCl. Therefore, the concentration of NaCl contacting the macula densa is abnormally high. In response to the elevated NaCl concentration, the macula densa cells release ATP, which functions as a vasoconstrictor. This causes a dramatic constriction of the afferent arteriole, which restricts blood flow into the glomerulus. This effect decreases the NFB by decreasing GHB, and therefore also decreases GFR. This allows more time for the absorption of NaCl by the renal tubule. Conversely, when the macula densa cells are exposed to slowly flowing filtrate with a low concentration of NaCl, ATP release is inhibited, causing vasodilation of the afferent arteriole. This allows more blood to flow into the glomerulus, thus increasing the NFB and GFR. In summary, the autoregulatory mechanisms maintain a relatively constant blood flow through the kidneys over an arterial systolic pressure range from about 80 to 180 mm Hg. Consequently, large changes in water and solute excretion are prevented. However, The intrinsic controls cannot handle extremely low systemic blood pressure, below 80 mm Hg, nor blood pressure exceeding 180 mm Hg. Extrinsic mechanisms are evoked in emergency situations such as an extreme drop in blood pressure systolic less than 80 mm Hg or extreme increase systolic greater than 180 mm Hg. The goal of the extrinsic controls is to return blood pressure to safe limits. It does this by increasing or decreasing glomerular filtration rate. Remember, the body is in trouble, so the goal has shifted from trying to maintain a relatively stable GFR of 120 to 125 ml/minute to altering the GFR in order to adjust blood pressure. We have two major mechanisms for doing this. They involve the sympathetic division of the autonomic nervous system working along with the hormone epinephrine released from the medulla of the adrenal gland, and the renin-angiotensin mechanism. The sympathetic nervous system and adrenal medulla share a common embryological origin, and they work together in times of stress, such as the fight versus flight response. The sympathetic nervous system uses nerves to release the neurotransmitter, norepinephrine. This action is rapid. The adrenal medulla releases a similar chemical called epinephrine, also known as adrenaline, into the blood circulation where it behaves as a hormone. Both substances cause vasoconstriction of the afferent arterioles. When the blood flow to the glomerular capillaries is significantly decreased or even shut off, then glomerular hydrostatic pressure (GHP) drops along with net filtration pressure (NFP) and glomerular filtration rate (GFR). Norep and epinephrine also directly stimulate the JG granular cells to release renin, and indirectly activate the renin-angiotensin mechanism by causing the cells of the macula densa to release ATP, which acts to constrict the afferent arteriole as well. The renin-angiotensin mechanism is triggered by several types of stimuli. We already looked at two of these on the previous slide. In addition, a sudden drop in systemic blood pressure will directly cause the JG granular cells to release renin. Once in the bloodstream, renin acts as an enzyme to convert an inactive plasma protein called angiotensinogen into another form called angiotensin I. Angiotensinogen, by the way, is made by the liver hepatocytes, which is another good reason for being kind to your liver. It is always circulating about in the inactive form designated by the ogan at the end of the word, ready to spring into action when renin is released. Angiotensin I in the bloodstream reaches the capillaries of the lungs, where there is another enzyme called angiotensin converting enzyme, or ACE. ACE converts angiotensin I into its active form, angiotensin II. You may wonder, why are there so many steps involved in this process? Well, the body doesn't want to make any mistakes, so this process and many others is finely regulated, and can be turned off if necessary should the situation reverse itself. Angiotensin II acts in 5 ways to stabilize systemic blood pressure. You can guess from the name, angio equals blood vessel and tensin to tense or constrict, that one of these ways is to behave as a potent vasoconstrictor by causing the smooth muscle in the wall of arterioles, small arteries, to contract. This reduces the diameter of the lumens of the arterioles, which increases pressure. Think of a tube of toothpaste. When you squeeze it, you reduce the diameter of the lumen of the tube, and the increased pressure causes the toothpaste to squirt out. Secondly, angiotensin II stimulates the reabsorption of sodium in 2 ways, one by acting directly on the renal tubule to pick up sodium from the filtrate and put it back into the blood, and 2 indirectly by causing the release of another hormone called aldosterone. Aldosterone belongs to a category of steroid hormones called mineralocorticoids. It too is released from the adrenal gland, but from the outer part called the cortex. Aldosterone also causes the uptake of sodium. So, we have 2 ways of taking sodium out of the filtrate and putting it back into the blood. How is this going to help increase blood pressure? You know from studying osmosis that water follows salt. In addition, you also know this intuitively because when you eat a lot of salty food, your body retains water for a time until the proper adjustments can be made. Furthermore, you may know someone who suffers from hypertension, chronic high blood pressure, and they are usually put on a low sodium diet. When your body retains water, blood pressure increases. In the situation of a dramatic decrease in BP out of the typical range, our body works with all the tools it has to increase BP, and that includes retaining salt. Third, retention of sodium stimulates the hypothalamus to release antidiuretic hormone ADH and activates the hypothalamic thirst center. This results with water retention caused by the action of ADH on the DCT and collecting Duct. ADH causes the synthesis of aquaporins, and so water moves out of the collecting duct and is drawn via osmotic pressure, higher concentration of solutes, in this case, NaCl, back into the bloodstream. In addition, ADH stimulation of the thirst center in the hypothalamus causes the individual to drink more fluids. The outcome is that blood volume increases, and therefore so does blood pressure. Note, diuresis dia equals through plus uresis equals making water refers to urine output. Antidiuretic hormone therefore inhibits diuresis urine output. This helps the body retain water when dehydrated or when BP or fluid volume becomes too low. The fourth way that angiotensin II works is to increase fluid reabsorption by decreasing peritubular capillary hydrostatic pressure. The efferent arteriole also has smooth muscle in its wall. Angiotensin II causes the efferent arterioles to constrict. This causes the hydrostatic pressure in the peritubular capillaries to decrease, which favors the movement of fluid back into the peritubular capillary bed, thereby adding to blood volume. It also reduces GFR by decreasing the total surface area of the glomerular capillaries for filtration. This is accomplished by targeting the glomerular mesangial cells, causing them to contract. The result is that GFR is reduced, and so is the amount of fluid lost in filtrate/urine. The diagram above summarizes the different ways that the renin-angiotensin system acts to maintain homeostasis, blood pressure. Clinical note: abnormally low urinary output less than 50 ml/day, is called anuria. This condition may indicate that the glomerular blood pressure is too low to support filtration. Extremely high systolic blood pressure is dangerous in many ways: heart attack, stroke, and damage to arteries. When blood pressure increases above 180 mm Hg systolic, the thin walls of the atria of the heart are stretched. In response to the stretching caused by elevated blood volume or blood pressure, cardiac atrial cells release atrial natriuretic peptides ANP to counteract the effects. The ANPs cause dilation of the afferent arterioles and constriction of the efferent arterioles, in effect, more blood coming into the glomerulus, but a greater resistance to blood leaving the capillaries via the efferent arteriole. This elevates the glomerular hydrostatic pressure and increases GFR. Aldosterone secretion is inhibited, and so tubular reabsorption of sodium ions is also inhibited. These effects result in increased urine production and decreased blood volume and pressure. Essentially, the person increases the volume of urine released. The decrease in body fluid volume lowers the blood pressure. This figure summarizes the physiological mechanisms regulating glomerular filtration rate GFR in the kidneys. Note that while the extrinsic controls are aimed at maintaining blood pressure, they also maintain GFR, since blood pressure drives filtration in the kidneys. Many important small substances such as glucose, amino acids, and water, are filtered in the renal corpuscle and enter the filtrate. If they stayed in the tubular fluid, they would be lost in the urine. Tubular reabsorption prevents this from happening. Needed materials in the initial filtrate are returned back to the blood. In order for this to happen, the solutes must pass through three barriers: 1. The apical luminal plasma membrane of the proximal convoluted tubule cells, 2. The basolateral plasma membrane, and 3. The endothelium of the peritubular capillaries. Our total plasma volume filters into the renal tubules about every 22 minutes. Therefore, all our plasma would be drained in the urine if not for tubular reabsorption. Tubular reabsorption is a selective transepithelial process that begins when the filtrate enters the proximal convoluted tubules. Recall that the PCT is lined with simple cuboidal epithelium with microvilli forming an extensive brush border. This increases surface area for reabsorption by 50x what it would be for a cell with a flat apical membrane. Because the tubule cells are connected by tight junctions, movement of substances between the cells' paracellular pathway is limited to water, and some important ions such as calcium, magnesium, potassium, and some sodium. The transcellular route consists of 4 steps: 1. Transport across the luminal membrane, 2. Diffusion through the cytosol, 3. Transport across the basolateral membrane, and 4. Movement through the interstitial fluid and into the capillary. Depending on the substance to be transported, the reabsorption process may be passive or active. Reabsorption of materials by proximal convoluted tubule cells involves passive transport, secondary active transport, and primary active transport. Sodium enters tubule cells downhill, passive from filtrate at luminal membrane and then is actively transported out of tubule cell by Na plus K plus AT phase pump on basolateral membrane. Sodium entry is coupled to transport of another solute secondary active transport. Active pumping of Na plus at the basolateral membrane creates concentration and osmotic gradients that drive 1. Reabsorption of water by osmosis. About 65% of water filtered is reabsorbed in the proximal convoluted tubule. This is because aquaporins are always present and osmosis is promoted by the medullary concentration gradient set up by the loop of Renal. This type of water reabsorption is called obligatory. The water is obliged to leave the tubule lumen because of the aquaporins and concentration gradient. 2. Reabsorption of anions and fat-soluble substances by diffusion, and 3. Reabsorption of organic nutrients and certain cations by secondary active transport symport with sodium. Note, most organic nutrients reabsorbed in the PCT move through the basolateral membrane by facilitated diffusion. Excluding sodium, there is a transport maximum Tm for nearly every substance that is reabsorbed. There is a finite number of carriers in the renal tubules available to carry each specific substance. If the carriers become saturated, then no more of the substance can be transported, and the excess spills over into the urine. This is what happens in diabetes mellitus. The table above summarizes the mechanism used by the cells of the proximal convoluted tubules to transport various molecules and ions. Beyond the PCT, the permeability properties of the remaining renal tubule changes drastically. In the loop of Renal, water reabsorption is not coupled to sodium reabsorption, and the permeabilities of the ascending and descending limbs are different. Water can leave the descending limb via osmosis, but solutes cannot. Just the reverse is true of the ascending limb where there is passive and active transport of solutes, but the ascending limb is impermeable to water. Water stays in, solutes transported out. These permeability differences play a vital role in the kidney's ability to form dilute and concentrated urine as is necessary to maintain homeostasis. By the time the filtrate reaches the distal convoluted tubule, only 10% of the originally filtered NaCl remains, and only 25% of the water that was filtered remains. In the DCT, reabsorption of Na+ and water depends of hydration status, blood volume, blood pressure of the body. This is called facultative water absorption, and is regulated by aldosterone. Na+ and ADH regulates water reuptake. Recall that PTH also targets the DCT of the kidney, preventing the excretion of Ca2+ when blood plasma calcium is too low. The diagram above shows a DCT cell with its various transport mechanisms for removing excess or harmful materials still remaining in the peritubular capillaries not removed by filtration. The materials are removed from the blood and secreted into the tubular fluid to be voided in the urine. Tubular secretion is important for 1. Disposing of substances, such as certain drugs and metabolites, which are tightly bound to plasma proteins and are not readily filtered. 2. Eliminating undesirable substances or end products of metabolism, such as urea and uric acid that were reabsorbed back into the blood. 3. Getting rid of excess potassium ions. Potassium is reabsorbed in the PCT and ascending loop of Renal. However, aldosterone targets the cells of the DCT and promotes active tubular secretion of excess potassium. 4. Controlling blood pH by secretion of hydrogen ions into the filtrate, and retention of bicarbonate, a base in the blood. The pH of the urine can vary over a wide pH range, however the blood pH is maintained within homeostatic balance. The collecting ducts receive tubular fluid from the distal convoluted tubules of many nephrons. The collecting ducts run through the medullary pyramids. There are two types of cells comprising the collecting ducts: intercalated cells which contain many mitochondria and microvilli, and principal cells. The intercalated cells play a role in maintaining the acid-base balance of the blood. The principal cells help maintain the body's water and sodium balance. Thus, the principal cells are also under the influence of ADH. When the body is dehydrated, decreased blood volume or blood pressure, ADH stimulates the incorporation of aquaporins in the collecting duct cells. Aldosterone targets the principal cells by causing them to activate or synthesize more Na+ and K+ channels. Therefore, little or no sodium leaves the body, so water is retained as well. The mechanisms that allow us to regulate the osmolarity and volume of the final urinary product with respect to changes in physiological conditions: maintenance of homeostasis, involve two countercurrent mechanisms which set the stage for the establishment and maintenance of the medullary concentration gradient. The mechanism for setting up the osmotic concentration gradient that makes it possible to control the volume and osmolarity of the urine as needed involves two countercurrent mechanisms. Notice in the picture above, that the osmolarity of the interstitial fluid in the renal cortex is 300 mosm, the same osmolarity as the blood. However, the osmolarity of the interstitial fluid increases deeper and deeper in the renal pyramid until reaching 1200 mosm at the apex. The two mechanisms responsible for the medullary concentration gradient are one countercurrent multiplication, and two countercurrent exchange. In countercurrent mechanisms, fluid flows through adjacent tubes in opposite directions. The countercurrent multiplier is the interaction between the flow of filtrate through the ascending and descending limbs of the long loops of Renal of juxtamedullary nephrons. The countercurrent exchanger is the flow of blood through the ascending and descending portions of the vasa recta blood vessels. It is the juxtamedullary nephrons with their long loops of Renal that create the medullary osmotic gradient. The filtrate entering the loop of Renal from the PCT is isosmotic to both blood plasma and the interstitial fluid of the renal cortex. The descending limb is permeable to water and impermeable to NaCl. Thus, as the filtrate flows down the tube, it becomes increasingly concentrated as water leaves by osmosis via the aquaporins which are always present. The result is that more water in addition to the 65% reabsorbed in the PCT returns to the blood. The osmolarity of the filtrate increases from 300 to 1200 mosm. The ascending limb is impermeable to water, but permeable to NaCl. Therefore, the filtrate becomes increasingly dilute as NaCl leaves, eventually becoming hypoosmotic to blood 100 mosm upon reaching the renal cortex. NaCl is actively pumped out of the tubular fluid as it ascends, because the fluid is becoming less and less concentrated. In other words, it's the active transport of NaCl by the ascending limb that sets the stage for the osmolarity gradient. Here's how the countercurrent multiplier works. Notice that there is a difference 200 mosm between the ascending and descending limbs at any given level. This reflects the ability of the ascending limb to pump out NaCl. Although a 200 mosm difference alone is not enough to produce a concentrated urine, countercurrent flow multiplies the effect of the small changes. This produces a gradient change along the length of the loop of 900 mosm, 1200 mosm, 300 mosm. The two limbs are close enough to influence each other's exchanges with the interstitial fluid they share. Water leaving the descending limb produces an increasingly salty filtrate that the ascending limb uses to raise the osmolarity of the medullary interstitial fluid. The vasa recta are the straight vessels that cling to the thin limb of the loop of Renal of juxtamedullary nephrons. The endothelial cells that comprise the walls of these vessels are physiologically an enigma because they allow free passage of both water and salt. This prevents the 300 mosm blood normally found in capillaries from washing away the medullary concentration gradient. What happens is that as blood flows deeper and deeper into the medulla, it loses water and gains salt, becomes hypertonic. Blood emerging from the medulla into the cortex picks up water and loses salt. This arrangement accomplishes the following: first, it delivers oxygen and nutrients to the simple squamous epithelial cells comprising the loop. Second, it protects the medullary concentration gradient by preventing rapid removal of salt from the medullary interstitial space and by removing reabsorbed water. Third, the blood in the bottom of the vasa recta is 4x the osmolarity of the blood everywhere else in the body. If this blood were to mix with the rest of the circulation, it would cause serious effects. Instead, by the time the blood leaves the loop of Renal, its osmolarity has been restored. Thus, the vasa recta function as countercurrent exchangers. They maintain the osmotic medullary gradient while removing reabsorbed water and solutes. Blood flow through vasa recta is slow, and the free permeability of the endothelial cells allows to water and NaCl permits passive exchange between the blood and interstitial fluid so that equilibrium is achieved. As blood flows deeper and deeper into the medulla, it loses water and gains salt, becomes hypertonic. Blood emerging from the medulla into the cortex picks up water and loses salt. This protects the medullary concentration gradient by preventing rapid removal of salt from the medullary interstitial space and by removing reabsorbed water. Recall, a solution's osmolarity is the number of solute particles/liter of water. The osmolarity determines the ability of the solution to cause osmosis. The osmolarity of blood plasma is 300 mosm. This is maintained by the kidneys by regulating the osmotic concentration and volume of the urine. Regulation of urine concentration and volume involves the cells of the DCT and collecting duct, and is dependent on the presence of the medullary concentration gradient when fluid must be conserved. We have all seen this mechanism work. Compare the volume and color of urine when you are very thirsty, hot day, exercise, etc. to the volume and color of urine when you have drank a lot of fluid. If your kidneys are working properly, you produce a small volume of densely concentrated urine as evidenced by the deep yellow color on hot days and when you are exercising and not replacing fluids. Conversely, on days that you have had a lot of fluid to drink, you produce a large volume of pale urine. These changes in urine color concentration and volume reflect the ability of the kidney to adjust plasma osmolarity and volume by controlling urine output. When you've had plenty to drink, the hypothalamus decreases release of ADH. The collecting ducts remain impermeable to water, no aquaporins present, so no water is reclaimed and put back into the bloodstream. If necessary, when the plasma osmolarity decreases, sodium can be removed from the filtrate by the DCT and collecting duct cells, so the osmolarity of urine can get as low as 70 mosm. The plasma and glomerular filtrate remains within normal range, 300 mosm, which was the goal. When an individual is dehydrated, the hypothalamus releases ADH in the amount necessary to maintain blood homeostasis. Certain neurons in the hypothalamus function as osmoreceptors. They can detect subtle changes in plasma osmolarity of as little as 6 mosm/L. These neurons respond to increased plasma and ECF plasma osmolarity by releasing antidiuretic hormone. The rate of release varies directly with osmotic concentration of the blood plasma. The higher the osmotic concentration, the greater the release of ADH. The principal cells of the collecting duct are the targets for ADH. When body is dehydrated, decreased blood volume or blood pressure, ADH stimulates the incorporation of aquaporins into the principal cells of the collecting duct. Water passes through the cells into the interstitial space and back into the bloodstream. Urine concentration may reach 1200 mosm, the concentration of the interstitial fluid in the deepest part of the medulla. With maximal ADH secretion, up to 99% of the water in the filtrate is reabsorbed and returned to the blood. Aldosterone targets the principal cells by causing them to activate or synthesize more Na+ and K+ channels. Little or no sodium leaves the body, so water is retained as well. Only 400 ml of highly concentrated urine is excreted. Note, aldosterone causes the reabsorption of sodium at the expense of potassium, which is secreted. Too much aldosterone can result in hypokalemia. To further illustrate how effective our kidneys are in regulating body fluid volume and osmotic concentration, consider that under normal conditions, urine volume is about 0.9% of the filtrate, and about 1.1 ml of filtrate is produced each minute. This equates to a total urine production of 1.5 liters/day, at an osmolarity that ranges between 600–1000 mosm/L. Under circumstances of severe dehydration, the urine concentration can reach 1400 mosm/L, and the urine volume represents only 0.2% of the filtrate, and about 1/4 of a milliliter of filtrate produced per minute, or only 400 milliliters in a day. Under the circumstances of overhydration, the osmolarity of the urine can range between 400 mosm/L to a little as 30 mosm/L. The urine volume represents 12.5% of the filtrate, and is produced at a rate of 16 milliliters/minute, or an incredible 22.5 liters/day. Diuretics are used to help individuals lose excess water. There are two major categories of diuretics. Osmotic diuretics are not reabsorbed in the PCT and thus carry water out with them. Examples of osmotic diuretics include the high blood glucose levels that diabetics suffer from. The glucose in the filtrate gives it a greater osmolarity than the interstitial fluid or plasma. Consequently, glucose pulls water out of the peritubular capillaries into the renal tubule. This explains why diabetics urinate and drink a lot. Alcohol works as an osmotic diuretic because it inhibits the release of ADH. Therefore, no aquaporins open up in the collecting duct, so the water is lost in the urine. Caffeine-containing beverages, coffee, tea, colas were thought to function as osmotic diuretics as well. However, recent evidence suggests that in order for them to have dehydrating effects, one must consume large amounts of it, more than 500 to 600 milligrams, the equivalent of 5 to 7 cups of coffee a day. I don't know, is 5 to 7 cups of coffee a day excessive? By whose standards? Another type of diuretics are called loop diuretics. This includes the commonly prescribed drug called Lasix. Loop diuretics work by inhibiting the formation of the medullary concentration gradient by acting at the ascending limb of the loop of Renal, stops active transport of NaCl. Have you ever heard of the expression, makes you pee like a racehorse? This expression came about because racehorses are given Lasix before they run in a race. A full bladder would slow them down. Renal clearance is the volume of plasma that is cleared of a particular substance in a given time, 1 minute. Renal clearance tests are conducted to determine GFR, which can be used to detect glomerular damage, follow the progress of renal disease, or determine renal clearance of drugs in order to prescribe the correct dose. Inulin, a polysaccharide, Mw equals 5000 can be infused into the patient. Alternatively, creatinine Rc equals 140 ml per minute can be used for a quick test because it is freely filtered. Because urine contains what the blood doesn't want or need, a simple urinalysis can yield lots of information regarding the health of the patient. Freshly voided urine should be clear. Cloudy urine indicates a urinary tract infection. The color can be pale to dark yellow, but it should be transparent. Urochrome, a pigment that results from the body's destruction of hemoglobin, gives urine its yellow color. An abnormal color such as pink or brown may be the result of eating certain foods, beets, or may be due to the presence of bile pigments or blood in the urine. Some commonly prescribed drugs and vitamin supplements can alter the color of urine. The pH of urine is typically slightly acidic, pH 6 to 6.5, however it can vary between 4.5 to 8.0 due to diet or changes in metabolism. Low pH reflects an acidic diet containing large amounts of protein. High pH could reflect an alkaline diet. The person may be a vegetarian, or prolonged vomiting, or a bacterial infection. Specific GR. Avidity of urine ranges from 1.001 to 1.035. The specific gravity of a solution is measured in comparison to the specific gravity of distilled water, which is taken as 1.000. Urine is water plus solutes, so a given volume of urine will have a greater mass than distilled water. The ratio of the mass of a substance to the mass of an equal volume of distilled water is its specific gravity. The chemical composition of urine is 95% water and 5% solutes. Urea is the largest solute component by weight. It is derived from the breakdown of amino acids. Uric acid is the end product of nucleic acid metabolism. Creatine is a metabolite of creatine phosphate. There are also varying proportions of different ions such as Na+, K+, P.O. 43, Ca2+, H+, HgO3, etc. The table above shows what should not be normally found in urine. Glucose usually does not appear in the urine, however, if a patient has consumed an excessive amount of sugary foods, they may present with glucose in their urine. This situation is non-pathological and will resolve itself. However, glycosuria can be caused by diabetes, and is in this case, pathological. Proteins in the urine also may be due to non-pathological causes such as excessive physical exertion, pregnancy and a high protein diet, but over 250 mg/day may indicate heart failure, severe hypertension, or kidney diseases. Ketone bodies occur in someone who has been seriously dieting or fasting, but are usually indicative of diabetes mellitus. Hemoglobin should not be in the urine, and neither should erythrocytes. This slide presents a great review diagram. It shows the steps involved with making urine filtration, reabsorption, and secretion. The solid red outline of the descending loop shows that water can leave the tubule via aquaporins and re-enter the blood, but solutes cannot leave. The green circles on the ascending limb indicate that NaCl is being actively transported out. The yellow circles indicate aldosterone-regulated sodium-potassium pumps. It shows that the DCT and the collecting duct are targets of aldosterone. ADH regulates the permeability of the collecting duct cells based on plasma osmolarity.